So I'm going to talk today about the main line of study of our lab at IDIBELL, which is to try to understand how cells die when we inhibit metabolism, especially glucose metabolism, with the aim to kill cancer cells by exploiting the particularities of the metabolism of cancer cells. So this is just an introduction of, a summary of the talk. I'll give an introduction a bit about apoptosis and a bit about cancer metabolism, and then I'll talk about two different lines of study in the lab. One is apoptotic death induced by glucose deprivation, and the other one is the use of glucose analogs against tumors, against sarcoma. So why do we care about glucose metabolism? Well, mainly because glucose is the main source of carbon and also of ATP for most cell types. So there's a lot of diseases that occur when there's a problem with glucose metabolism, such as diabetes, as everybody knows, but also lack of glucose is involved in diseases such as myocardial infarction and stroke. So we would like to know what determines sensitivity of cells to lack of glucose. On the other hand, this is a very fashionable subject at the moment because tumors have a high demand of glucose. This is what is known as the Warburg effect that was known in the 30s, but still it's again, it's a fashion at the moment because we're finding out so many things about tumor metabolism that there's a lot of interest. So in this sense, we're interested in understanding what type of tumors we can kill by inhibiting glycolysis, and also what determines sensitivity of tumor cells to anti-glycolytic drugs. And we're more interested in the apoptotic part, but we're also interested in upstream sensing signals. So this is a picture of how metabolism looked like until 10 years ago. What people were interested in was how what we eat is converted into small pieces. That's what we digest. And then from glucose, amino acids, and fatty acids, these compounds go to the mitochondria, these metabolites. Krebs cycle occurs, and a lot of ATP is produced by respiration. This was the focus. We're interested in how to digest and how to obtain especially ATP from glucose and from the other metabolites. Now, in the recent few years, we're understanding that the picture is completely different for proliferating cells, especially for cancer cells, but also for other proliferating cells. And what's emerging is that glucose is not only required for ATP in the mitochondria, but glucose is required for almost everything. So in your tissue culture medium, you have serum, but you don't have nucleotides. Nucleotides, for instance, are produced from glucose. Also, glutamine is very relevant, but I won't talk about it today. So glucose is required to make DNA and to make RNA. Glucose is required for glycolysis, but also from pyruvate and from some other intermediates here, cells make amino acids, which are non-essential amino acids, because cells can make them from glucose. Also, in tumor cells, this pathway is enhanced, and there's production of lactate in what we call aerobic glycolysis. And it's still not very well understood why, but lactate is supposed to confer some growth advantage to tumor cells. And also, keep in mind that there's no lipids in your culture medium in general. Tumor cells make lipids to make membranes and signaling molecules from glucose as well. So glucose is required for almost everything. So what happens when cells know that they don't have enough glucose? Cells sense it by these metabolic checkpoint proteins. This is a novel concept as well. When there's not enough amino acids in the culture medium, the protein mTOR is inhibited, so cells stop growing. And when there's not enough glucose, the ATP levels go down, and this activates the AMPK, AMP-dependent protein kinase, which turns on P53 and again inactivates mTOR. So the purpose of this checkpoint is to promote survival of the cell by enhancing pathways that will repair damage, but also that will make cells not use more nutrients that they have. So on one hand, repair pathways such as autophagy or Also fatty acid oxidation that can promote ATP generation. The ER stress signaling pathways is also engaged here, as you will see later. But also everything else is, every anabolic process is stopped. Protein synthesis stops, DNA synthesis stops, and in general, all anabolic processes. But if something goes wrong here, and this is very important because this is the novel concept, if the metabolic checkpoint is wrong, for instance, say cells don't have p53, as is very common in tumor cells, or they cannot activate AMPK, or mTOR is hyperactivated, then something's wrong here. The cells, instead of setting up this program, they undergo cell death. So this is what is making tumor cells more sensitive to lack of nutrients. It's not only that they proliferate more, so they need more nutrients, but it's also that they cannot undergo the proper checkpoint, and then they continue growing without knowing that they don't have the resources to grow. So we're interested in this part of the picture, which is how do cells die. And it was described that cells die, especially hematopoietic cells, that was the only thing that was really known, die by apoptosis. And this is mediated by some BH3-only proteins that I will tell you about later. And some non-hematopoietic cells die by necrosis. And this is probably mediated by reactive oxygen species, but we don't know much about this. So what is apoptosis? This is, I just have a small video for people that haven't seen this in culture. You have to be very lucky for not seeing this in your culture. Cells shrink during apoptosis when they receive a signal. Cells shrink, they bleb, the membrane starts blebbing. And cells end up detaching, and at this moment you should not be able to detect this in vivo because phagocyte would have arrived and would have removed the apoptotic bodies. So the cell is gone very quickly, and the purpose of apoptosis is to not cause inflammation in the culture. If cells die in a different manner, they usually release their content and inflammation occurs. Now, just a brief update of what we understand by apoptosis recently. We define it by morphological and biochemical criteria, but all of these morphological and biochemical events are depending on caspases. So we currently define apoptosis as caspase-dependent cell death. Caspases are proteases that disassemble the cell, but not by chopping everything like trypsin or chymotrypsin or other proteases would do. It's by cutting substrates in some specific regions, and these substrates have specific roles during apoptosis, but mostly the cell disassembles. So it's not really programmed cell death because there are other forms of programmed cell death, and it's not programmed when there's an accident and cells die by apoptosis. For instance, you get burned and some cells would die by necrosis, but some others would die by apoptosis. That's not really programmed. But it's organized by a genetically encoded program. That's correct. So as I said, caspases are the proteases that carry out the process. In the microscope, in the fluorescence microscope, you can follow some steps. During the phase of stimulus, lots of things are happening, but you don't see anything. This is cytochrome C GFP in the mitochondria of the cells. In the decision phase, when the decision has been made, then mitochondria become permeabilized, and this happens because of regulation of BCL2 family members in one of the pathways at least. During the execution phase, now the cell is doomed. There's caspase activation, and this causes a lot of morphological events like membrane permeabilization and annexing exposure. In this case, the pink is the annexing binding to the membrane. Sorry, phosphatidylserine exposure is a lipid that is not supposed to be there. Then annexing binds to the lipid, so cells become annexing positive. And DNA is chopped. In this case, DNA is in blue, is stained with propydium iodide. It's all over the cell because it's chopped in very small pieces. Biochemically, there are two main pathways with the same outcome. The two main pathways are the extrinsic or death receptor mediated and the intrinsic or mitochondrial pathway. They are engaged by different signals, and the extrinsic of death receptor pathway is mostly important for the immune system. It's a cell, the immune system uses this pathway to kill infected cells or superfluous cells, but mostly to kill infected cells. And it's engaged by TNF, trail, or first ligand, which are the death ligands. And it's very simple because there's a recruitment of caspases to the same complex that is formed in the membrane. So the death receptors aggregate in the membrane when they are engaged by a ligand, and caspase-8, which is an initiator caspase, is brought up to the membrane, and just by oligomerization, caspase is activated. There's a lot of regulation, protein regulation at this level, but these are the basic essential components. When this caspase is active, executioner caspases, such as caspase-3, are active, and then cells die. Now, everything else that is not a dead ligand engages the mitochondrial pathway. It's called intrinsic pathway, but of course it's also engaged by extrinsic stimuli like growth factor removal or drugs. Extrinsic will be growth factor removal. The rest will end up doing that damage intracellularly. So these stressors cause the regulation of BCL2 family proteins that are the ones that regulate the mitochondrial permeabilization. Then caspase-9 is formed in the apoptosome, and this again triggers the activation of executioner caspase. In a little bit more detail, what you see about how initiator caspases are active in the extrinsic pathway, they're activated by bringing up pro-caspase-8 to the death receptor. This is inhibited by a molecule called FLIP. I'll talk a bit about it later. And this caspase then becomes active by cutting itself, and this releases the active caspase into the cytosol, and this activates the executioner caspase. In the mitochondrial pathway, caspase-9 is activated in a complex which is kind of similar to this, which is the apoptosome. The critical point in the mitochondrial pathway is then mitochondrial permeabilization, and this is achieved and controlled by BCL2 family proteins. You've probably heard about these proteins. Plenty of them are mutated or overexpressed in cancer, like BCL2, MCL1, and BCL-XL. And some proteins of this family, such as BAX and BAC, are responsible for making a pore in the outer mitochondrial membrane, and this causes the release of proteins from the intermembrane space and the activation of caspase-9. The anti-apoptotic proteins are usually or can sequester the pro-apoptotic proteins. And then there's a three group of members of this family called the BH3 family, the BH3 only proteins, that what they do is to either neutralize the anti-apoptotic, so this would release BAX and BAC to make the pore, or directly activate BAX and BAC to form the pore, or both. So ultimately, I mean, what's more upstream of the pathway and where more the most effort is devoted now in researching apoptosis is understanding how BH3-only proteins engage this process and which drugs trigger activation of what BH3-only proteins. There are other forms of cell death, as I've mentioned. Necrosis is basically everything that we cannot define. All forms of cell death that happen when that is not apoptosis. Now, autophagic cell death is very popular at the moment, so when autophagy is overactivated or nobody knows exactly how, but autophagy engages a form of cell death that is not apoptosis. Necroptosis is a form of necrosis that is mediated by a protein called RIP1 and RIP3, and well, this is not that relevant. I just want to point out the method that we use mostly in the lab to analyze apoptosis and to distinguish it from necrosis. This is a profile of cell cycle of cells that have been fixed when we incubate them with propylium iodide. Cells in G1 are here, cells in S are here, G2 are here. So everything that's left is cells in sub G1. These cells have degraded DNA and they have less DNA content. It's not the only reliable method. You need to do a lot of things if you're trying to understand whether your cells are dying by apoptosis or necrosis, but at least this is very handy because you can fix the cells and run them through the fax any other day. Other methods are looking at chromatin condensation, annexing, and caspase cleavage. So going back to the pathway that we're interested in, we were trying to analyze how cells die when you deprive them of glucose. For the interest in cancer, but also in ischemic diseases. So we used a model in which the mitochondria was completely inactivated, the mitochondrial pathway, not the mitochondria are happy, to be able to study this necrosis that is supposed to be occurring during ischemia in several tissues, and also because the inactivation of the mitochondrial pathway is very common in tumors. So we employed cells that are deficient in BAX and BAC, so they can undergo mitochondrial permeabilization, and they have to die by necrosis, we thought, because the mitochondrial pathway is inactive. They can still die by the other pathway, the death receptor pathway, the one that engages caspase-8, because that pathway is totally fine and they don't require the mitochondria, right? So to our surprise, MEFs that are deficient in BAX and BAC died by apoptosis. And by apoptosis, I mean that when you remove glucose. We didn't expect this. We expected necrosis because the glucose deprivation had been shown to kill cells either by apoptosis, mitochondrial apoptosis, or necrosis. So when you incubate the cells with caspase inhibitors, they are kept alive even though they detach, but they are alive in the sense that they don't incorporate propydium iodide, the membrane is intact. This is the kinetic of cell death measured also by sub-G1 DNA degradation, which is a hallmark of apoptosis. It's the same kinetic as with propydium iodide. And caspase inhibitors such as CBAT, QBD, and a bunch of other caspase inhibitors block cell death, meaning that it's apoptosis. It's caspase-dependent. Now, if you see the kinetic of the wild-type MEFs, they die a lot faster than double knockouts at BAX-BAC deficient MEFs. This is the cleavage of caspase-3, which is another hallmark of apoptosis, because when caspase-3 is activated, it's cleaved, and you can detect the smaller fragments with an antibody. And this is looking at the caspase substrate. We did a lot of tests to demonstrate that this was apoptosis, because it was very difficult to believe that cells that have the mitochondrial pathway neutralized, they can still die by apoptosis. So the only thing that wasn't making sense, and I just talk about it as a curiosity, is the nuclear condensation, which is not classical apoptotic nuclear condensation. This is how normal nucleus would look like when you transfect cells with histone to be GFP that binds the DNA. Apoptotic cells, like these same cells when they are treated with TNF through the non-mitochondrial pathway, they die totally normally by apoptosis. They condense the chromatin. But when we incubate these cells without glucose, or the same in the presence of caspase inhibitor, the DNA degradation is different. We don't know what this means, but it's not the classical chromatin condensation, and we think this might have led some people to conclude that cells die by necrosis just because the condensation is not apoptotic condensation. We're not sure of this, but we're studying this. Well, we're not studying the condensation, but we're trying to understand whether there's really necrosis or not. This is how cells that are completely dead look like when they're killed by something that induces necrosis, Tapsic argon. But the question was, biochemically, how is it possible that you induce apoptosis in BAX-BAC deficient MEF? So after a lot of tests to demonstrate that the mitochondrial pathway is totally inactivated, that there's not a problem with our model, that there's not another possibility of an alternative mitochondrial pathway, that caspase-9 is absolutely not involved, we turn to the other initiator caspase, which is caspase-8, that is the one that is engaged by death receptors normally. And indeed, caspase-8 was the initiator caspase in glucose deprivation-induced apoptosis. This we can show by several means. I show you here, for instance, this is the kinetic of cell death. And in red, there's the cells that have been infected transfected with a retrovirus, which encodes for a viral protein, CRAM-A. CRAM-A is a protein that inactivates caspase-8 and caspase-1. This is a natural protein that occurs in viruses that try to inactivate caspase-8 to evade the immune response. So when we transfect the cells with CRAM-A, we get the same degree of protection that when we incubate with caspase inhibitors. Also, when we knock down caspase-8 but short herping or siRNA in transient, we see cell death is prevented, and cleavage of caspase-3 is also prevented, and of another substrate of caspase-8, which is called BID. And trying to understand how caspase-8 could be activated by a stressor that should engage the mitochondria, we thought of plenty of things, like, for instance, maybe cells were producing TNF or were producing death ligands, and this was killing the cells in an autocrine fashion. We blocked every possible death receptor, death ligand, nothing happened, so this is not the case. And actually, we checked for production, and they were not produced. So we tried to at least see whether death receptors were involved. Maybe there was an intracellular aggregation of death receptors that is activating caspase-8 in the absence of the ligands. And to do this, we overexpress FLIP, which is a molecule that is very similar to caspase-8, but it competes with caspase-8 for binding to the receptor, so it doesn't allow for the formation of the complex. So when we overexpress FLIP, cells are completely protected from death ligands, as you would expect, completely protected from TNA and from antifaz, but they are absolutely not protected from glucose deprivation. So we have a form of activation of caspase-8 that is independent of the classical death inducing signaling complex. So we haven't solved this puzzle yet, but the latest thing that we tried to analyze was to see whether caspase-8 was being activated in a fashionable complex now, which is called the riboptosome, that can be formed by some drugs. It's been very recently described in Molecular Cell, but it was also described last year in response to TNF and SMAC mimetics. And this complex is formed by a kinase called RIP1, and it's composed of caspase-8 and some other molecules. So we tried to analyze whether caspase-8 was activated in this complex by inhibiting the activity of this kinase, RIP, by two different means. One was by using a compound called necrostatin, which is the inhibitor of the kinase, and the other one was by knocking down RIP. So in these totally parallel lines, you can see cells treated or non-treated with necrostatin when deprived of glucose, so the necrostatin doesn't do any effect. There's not depending on RIP kinase activity. But RIP is also not forming a scaffold that would activate caspase-8, because when we don't regulate RIP kinase enough to prevent death induced by something else that forms the apoptosome, again, cells are completely parallel. RIP has nothing to do with this form of cell death. Or you need so little amounts of RIP that we cannot observe any effect of RIP knockdown. So again, so far we haven't been able to identify this, and we have some ideas, but we've tried all the usual suspects, so we don't know at the moment. But what we have clearly found is another form of activation of caspase-8, again unknown, that is formed when stress that should induce the mitochondrial pathway that encounters cells that cannot undergo the mitochondrial pathway. And glucose deprivation is one of such stressors, and there are more that we're not studying because it's not enough people. We wanted to know, however, that this was not really something funny happening in back deficient MEFs that are difficult to find in nature. And we turned to HeLa, which are the common laboratory cells that we used, and it turns out that HeLa that are transfected with a mitochondrial what's an inhibitor of the mitochondrial pathway, such as BCL-XL, they are protected, they are not protected from glucose deprivation. They die with the same kinetics that regular HeLa, right? But death is apoptosis, so it's not the mitochondrial pathway. We know that these cells for sure are incapable of undergoing the death through the mitochondrial pathway, and they still die by apoptosis. But moreover, regular plain HeLa, not the ones that have the mitochondrial pathway forcibly inactivated, but plain HeLa, die in a caspase-8 dependent manner. There's a necrotic component here. Caspase inhibitors don't prevent death completely, but knocking down caspase-8 is the same as using caspase inhibitors. So normal plain HeLa die in a caspase-8 dependent manner when deprived of glucose. So we have added a little bit to the picture here of how cells die when cells are deprived of glucose. And some cells undergo mitochondrial apoptosis, hematopoietics as far as we know. Some cells undergo necrosis, and some cells undergo caspase-8 mediated apoptosis that we picture here with a strange DNA condensation. Now in the second part of the talk, I'll talk about the more recent work, which is try to understand how tumor cells die when we give them a glucose analog that doesn't allow them to make glycolysis. It's not the same thing, especially we know now, it's not the same thing to deprive glucose than to administer something that competes with glucose and that inhibits glycolysis. I'll show you what we know about this. But especially we don't want to remove glucose from a patient because that's kind of complicated. But we can give patients anti-glycolytic drugs such as 2-deoxyglucose. We're mostly interested in sarcoma because is the lab we're collaborating with. Oscar Martinez Tirado works in several types of sarcoma, and we thought this was interesting to test because really sarcoma are very difficult, tumors that are very difficult to treat and are understudied. We're focusing mostly in rhabdomyosarcoma, which are pediatric tumors. Some people work here in rhabdomyosarcoma, by the way. The most common sarcoma that are very aggressive and very resistant to chemotherapy. There are two major types, the embryonic and the alveolar. But we're also interested in other soft tissue sarcomas, such as Leo, Ewing, and liposarcoma. So the drug that we would like to use, this is the glycolytic pathway. Don't worry, I still don't know it after so many years. But just to let you know that we can inhibit the glycolytic pathway at plenty of steps. There are drugs that target the glycolytic pathway. After all these years, people have developed all sorts of inhibitors. Some are very toxic to people, as you can imagine. But some have been, like such as 2-deoxyglucose, which is the one that we use, are surprisingly well tolerated by patients. And there's clinical trials right now ongoing in the USA to treat pancreatic tumors, and it has been given several times, although the results were not spectacular, but we don't know whether it's because they didn't hit the right type of tumors. Maybe that's the case. So 2-deoxyglucose blocks the pathway very high up. It competes with glucose, but it also gets trapped into the cells, gets phosphorylated, and at this point cells cannot use the glucose anymore because it competes with glucose for the rest of the pathway, so it's not metabolized. So first of all, we wanted to make sure that sarcoma are glycolytic tumors because not every tumor is so dependent on glycolysis. So we took these cells and we deprived them of glucose as we were doing. And we saw that finally, yes, some cells do die by necrosis when you deprive them of glucose. And for instance, these types of rhabdomyosarcoma, this is the type of cell death that you would see, the amount of cell death that you would see if you analyze sub-G1 DNA degradation. Looks like cells aren't dead, but if you look at them in the microscope, they are dead. And indeed, if you put propydium you see that the kinetics are completely different. And death is not prevented at all by using caspase inhibitors. So this is what we would call necrosis. Same thing, if you look in the microscope, staining the cells with propydium iodide or trypan blue, they are very sensitive to glucose deprivation, and this is not prevented by QVD. So cells, rhabdomyosarcoma and other types of sarcoma are very sensitive to glucose deprivation, and they die by necrosis. So we decided to see what happens when you treat them with 2-deoxyglucose, and this is a crystal violet curve in which you can see that the different doses of 2-deoxyglucose cells stop growing, but they don't only stop growing at a bit higher doses that are a bit higher, they also die. Now this was a bit shocking because we were analyzing here sub-G1 DNA degradation, that is a apoptotic marker, and these are exactly the same cells that I have shown you before that die by necrosis when you deprive them of glucose. So it's not the same, and if somebody here is working with 2-deoxyglucose as a low glucose mimetic, as plenty of people do when they study ischemia, be careful because it's not the same. 2-deoxyglucose and glucose deprivation are doing different things. And death is completely prevented by a caspase inhibitor. So we know that this is apoptosis. 2-deoxyglucose doesn't only induce apoptosis in rhabdomyosarcoma, in alveolar rhabdomyosarcoma. It also induces differentiation of cells, what looks like terminal differentiation. And this is interesting for therapy because even if cells aren't killed, at least they cannot proliferate anymore. So these rhabdomyosarcoma are coming from muscle-like type of cells, like a skeletal muscle. So in the presence of 2-deoxyglucose, they turn back, or they change into something that look like muscle fibers. And also, these tumors are generated by a chromosomal translocation. It's like a BCR-ABLs, and this is a chromosomal translocation, which is PAC-FOXO. And 2-deoxyglucose promotes the downregulation of the chromosomal translocation. We're not sure what this means yet, but it's possible that this is responsible for the differentiation phenotype, but we don't know whether it has anything to do with cell death. Unfortunately, we have tried other types of sarcoma. Everything that I've told you about death is alveolar rhabdomyosarcoma, but we've tried embryonic rhabdomyosarcoma, several cell lines, and they're completely resistant to apoptosis, only at super high doses, which is something. And even though they stop growing, these are cells that have been grown for 72 hours, and these are cells that have been grown with a high dose of 2-deoxyglucose. They do stop growing, this crystal violet, but the moment you wash it away, cells continue growing away. They're arrested, but they're not senescent. They don't really care, and we don't understand the difference. We've done some experiments, but we don't understand. Same with a bunch of liposarcoma, bunch of leiomyosarcoma, and unfortunately, the master student at this point was desperate. We decided to study the cells that do die with 2-deoxyglucose to try to understand the mechanism, which are the alveolar rhabdomyosarcoma. So again, a reminder of the two pathways of apoptosis. We're trying to understand what are the key molecules here that could predict whether tumor cells will be responsive or not to death induced by 2-deoxyglucose, and hopefully by all sorts of antiglycolytic analogs. So the extrinsic pathway, which is mediated by caspase-8, that is engaged by glucose deprivation at least in a couple of cells we've studied before. The mitochondrial pathway, which is the classical apoptotic pathway that is regulated by BCL2 family members, and they both converge in activation of caspase, executional caspases. So we made sure that 2-deoxyglucose is inducing apoptosis. We're doing now the other way around that before. We really want to make sure that this is apoptosis, and indeed we saw cleavage of caspase substrates and caspase activity by measuring peptidase activity in the fluorimeter, which is inhibited by a caspase inhibitor. And again, contrary to what other people would do, we started first with by looking at the caspase-8 pathway instead of the mitochondrial pathway. And we found something funny, I don't know what to make of this, that the sensitive cells are the ones that don't express caspase-8. The resistant cells, like embryonic rhabdomyosarcoma, do express caspase-8, at least when we're comparing with HeLa cells and with other types of cells. We don't understand it, but some epigenetic silencing has been found in pediatric tumors, some epigenetic silencing of caspase-8. But it's not the caspase-8 mediated pathway. These cells are completely resistant to death receptor engagement. So we turn to look at the mitochondrial pathway, of course. And as a reminder of the key molecules in the mitochondrial apoptotic pathway, we have the pro-apoptotic pore-forming protein that are BAX and BAC that are absolutely essential for pore formation and for cell death. The anti-apoptotic ones that are BCL2, MC1, BCL-XL, them are the most studied ones. There are more. And the BH3 only are the ones that come and activate BAX and BAC and inactivate BCL2 and the anti-apoptotics. So we've studied, we made sure that this is the mitochondrial pathway by analyzing translocation of BAX and BAC or activation of BAX and BAC in the mitochondria, which can be detected by some antibodies that detect the exposure of one epitope that only happens when these proteins are inserted in the mitochondrial membrane and so are active. And we detect that cells that have classical chromatin condensation also have BAC and BACs active in the mitochondria are reactive to this antibody. And also overexpression of the anti-apoptotic proteins of the mitochondrial pathway, such as MC1 and BCL-XL, when we transfect the cells with higher doses, high doses of plasmid encoding for these proteins, they are protected. They protect from 2-deoxyglucose. The same the other way around. If we downregulate MC1 or BCL-XL, cells are more sensitive to 2-deoxyglucose. Now, trying to analyze levels of these proteins to see if we could get a clue of which ones were the important ones for cell death. BAX and BAC don't change. Now, the anti-apoptotic, funnily enough, BCL2 and BCL-XL go up a lot during cell death, and we don't understand why, because these cells are still sensitive to 2-deoxyglucose. But the one that does go down is MC1, and this is not a surprise because MC1 has been shown to be downregulated by glucose deprivation by a group we're collaborating with. So we thought that MC1 was playing a role in cell death, but the one that we're more interested in is the BH3 only proteins, the ones that are upstream and that are which are the ones that are activated by glucose deprivation, by 2-deoxyglucose. And we focused on NOXA, BIM, and PUMA because these ones have been shown to be induced by glucose deprivation in hematopoietic cells. So the treatment with 2-deoxyglucose did induce NOXA and BIM, even though NOXA had very long time points when cells are very dead. PUMA is not induced, it's a p53 target protein, and our cells are deficient in p53. So we knock down both NOXA and BIM, or both combined, and downregulation of NOXA protects from cell death, downregulation of BIM doesn't, and in a short time points it even sensitizes. And downregulation of NOXA and BIM combined doesn't make more than just downregulating NOXA. So NOXA is the BH3 only protein that is critical for cell death induced by antiglycolytics. Trying to understand the regulation of NOXA and MCA1, why is NOXA going up, MCA1 is going down. We looked at regulation of the messenger, transcription, and we couldn't find any transcriptional regulation. Now, both of these proteins are very, very sensitive to inhibition of translation. They're very short-lived proteins, but especially MCA1. But both of them are very rapidly induced when you use proteasomal inhibitors, for instance, and actually NOXA is responsible for death induced by bortezomib. So what we see is that if we incubate the cells in the presence of cycloheximide, both of them go down very quickly. And after treatment, 48 or 72 hours of treatment, both proteins are stabilized. And this doesn't make a lot of sense because MCA1 goes down. How can a protein that is stabilized after a treatment be decreased if it's not at transcriptional level either? Then we realized that people had described general inhibition of translation when glucose is not present. Of course, mTOR is inactivated. And indeed, the same thing happens with 2-deoxyglucose. The mTOR pathway, this is a downstream substrate of TOR. mTOR pathway is inactivated, and also the initiator EIF2 pathway of translation is also inactivated. We see this by phosphorylation. So translation is inhibited. And this is probably what is causing MCA1 downregulation. But we wanted to go up. Like what event, what is engaged by 2-deoxyglucose that is different from glucose removal, that is causing MCA1 and NOXA regulation and cell death? So if you remove glucose, there's a lot of things that happens to the cells. They don't have lipids, they don't have DNA, they make reactive oxygen species, they don't have enough proteins, they crash. Now, there was something that was described that was different with 2-deoxyglucose and glucose deprivation. And it's something very important that people never include in these graphics, and that is that glucose is required for protein glycosylation. Of course, protein and lipid glycosylation. If you don't have glucose, you don't glycosylate anything. So, and glucose and glucose deprivation and 2-deoxyglucose have been shown to have slightly different effects on glycosylation profile. So we decided to use a sugar that is mannose that can bypass the possible block of 2-deoxyglucose to the glycosylation pathway. So it turns out that when we use mannose, cells are completely protected from death induced by 2-deoxyglucose. This gives us this is telling us that glycosylation is inhibition of glycosylation is more important than all the things that are happening in the cell in response to 2-deoxyglucose. But to make sure that mannose was not preventing 2-deoxyglucose uptake or was not being metabolized by some strange pathway, there's not really a lot of information about metabolism of mannose in human cells. We added mannose, and we looked at the loss of ATP induced by 2-deoxyglucose. 2-deoxyglucose promotes a loss of ATP, of course, it inhibits respiration in the end. And the presence of mannose did not alter the loss of ATP, did not rescue the loss of ATP. This is telling us that it's glycosylation and not really the other metabolic effects of 2-deoxyglucose that is killing the cell. So what's happening to the cells when you inhibit glycosylation? The first thing that happens is that the cells accumulate in the endoplasmic reticulum because they cannot fold properly. So it's endoplasmic reticulum stress. And to make sure this was the case, we looked at endoplasmic reticulum markers, such as the GRP proteins and the transcription factor ATF4. that is induced very rapidly after ER stress, endoplasmic reticulum stress. So in the presence of mannose, ER stress induced by 2-deoxyglucose was completely prevented. But not only that, downregulation of MCA1 was completely prevented, upregulation of NOXA was completely prevented, and BIM, so-so, we're not sure why, because BIM is usually induced via ER stress. It's a very good target of ER stress protein. But this was not the case, it was only partially prevented. So another proof to make sure that it was the problem with glycosylation and also the ER stress that was causing cell death, we employed a classical ER stress inducer such as tunicamycin, which is an inhibitor of glycosylation. And tunicamycin also produces a downregulation of MCA1 and the late upregulation of NOXA when we use a dose that is killing with the same kinetics as 2-deoxyglucose. So we're fairly convinced that it's ER stress what is causing death of the cells. But to prove it even further, we turned to study one of the ER stress pathways that had been shown to be induced by lack of glucose. And this is the PERK pathway. I don't know if you're familiar with the ER stress. I don't have an introduction slide. But the PERK IF2 pathway is engaged when there are problems by glycosylation. And one of the first proteins that is induced is this transcription factor ATF4. This transcription factor has been shown to induce NOXA. So we thought, we knock it down, NOXA is not induced, cells don't die, and this is the pathway. And we knocked it down, and we prevent cell death, indeed. But to our surprise, NOXA levels didn't change. And indeed, at this point, we already knew that NOXA was not induced transcriptionally. But what the knockdown of ATF4 is doing is it's maintaining MCA1 levels. Somehow, we don't understand why, but knocking down ATF4, even in untreated cells, just by being with the siRNA for a long time, they're happy and they don't downregulate MCA1, and death is reduced. So at this point, we have a pathway in which 2-deoxyglucose inhibits glycosylation. This produces ER stress. ER stress induces the PERK ATF4 pathway, and somehow this leads to the upregulation of NOXA, especially to the downregulation of MCA1, and cells undergo mitochondrial permeabilization and they die. So as a conclusion, I've told you that glucose deprivation and 2-deoxyglucose induce different forms of cell death in the same cell lines. That glucose deprivation can promote activation of caspase-8 in an atypical complex that we haven't identified yet. 2-deoxyglucose is effective against tumors that are very difficult to kill, and with some 2-deoxyglucose is in clinical trials, and we hope we can continue with these experiments and end up in a clinical trial. 2-deoxyglucose induces apoptosis through the mitochondrial pathway. NOXA and MCA1 are key proteins involved in the mitochondrial pathway. And it's ER stress rather than the inhibition of the ATP synthesis that is causing apoptosis. So with this, I'd like to finish, thanking especially Silvia, which is the one that did most of the work, and Oscar and our other collaborators at IDIBELL, and Alfredo that did the first part, the experiments of the first part. And thank you very much.